Everyone, I am Dr. M. Parimala, Assistant Professor, PG and Research Department of Microbiology, St. Joseph's College of Arts and Science, Autonomous Cuddalore, Tamil Nadu. I am going to present a topic two-dimensional electrophoresis. Let's see the introduction about the two-dimensional gel electrophoresis. It is abbreviated as 2DE. It is called two-dimensional electrophoresis, a powerful tool for proteomics work. It is used for the separation of complex protein mixture from biological samples. The two-dimensional chromatography separates the protein molecules based on two separate steps. Now let's see what are the two major steps. The first step is called the separation of the molecules based on isoelectric focusing. It is abbreviated as IEF, which is used to separate the protein according to their isoelectric point, PI. The second step, it is the sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is abbreviated as STS page, which is used to separate the proteins based on their molecular weight. So thousands of proteins can be separated and the information about their isoelectric focusing as well as the molecular weight of the protein molecules can be studied. The two-dimensional chromatography, it is a widely used method for protein analysis which has the ability to separate the proteins. It can provide direct visual information of changes in protein like post-translational modifications. It can be also used to other analysis such as proteome analysis and detection of biomarkers, drug discovery and so on. Now let's see how the sample can be prepared. The sample need to be converted to a physico-chemical state that is suitable for the first dimension of this 2DE, that is called isoelectric focusing, and keep the native charge of the constituent proteins. In order to get the very good result, the appropriate sample preparation has to be made. The sample preparation is various because of the different in the type as well as the origin of the proteins. Ideally, the process will result in the complete solubilization, disaggregation, denaturation and reduction of proteins present in the sample. Now let's see the first dimension of this two-dimensional electrophoresis, and it is said to be isoelectric focusing. The first dimension is to find out the isoelectric point for the proteins. The proteins are generally amphoteric molecules, that is, they may possess positive, negative or zero net charge. So they carry those charges, it is mainly depends upon the pH of the surrounding medium. The isoelectric point, that is the pA, it is defined as the pH of a solution at which the net charge of the protein becomes zero. So either it may be positive or negative based on the pH gradient, it will become zero charge. A protein with a positive net charge will migrate towards the cathode, become less positively charged until it reaches its isoelectric point. While a protein with a negative net charge will also migrate towards the anode, that is the oppositive charge, become less negatively charged until it also reaches its isoelectric point. In this figure, you can see the first dimension of this 2D electrophoresis, that is the method for detecting the isoelectric focusing, that is the isoelectric point of the protein sample. So in this, we have to load the protein samples, that is the mixture of protein, usually at the high pH. That is, if the pH is high, it is basic. If the pH is low, it is said to be acidic. So in order to find out the isoelectric focusing, that is in a pH gradient gel, we are adding the sample at the mixture of the protein at the basic region and we are applying the electric field. So since we are applying the electric field, the molecules will start to migrate, and it depends upon the size of the molecule as well as the charge carried by the molecule. They will move at the different rates. The larger molecules are usually lagging behind even though it will be able to move and it will reach based upon its charge. So we will see the separation of the molecules, that is based on the charge as well as the size. So the protein mixture, it is loaded at the basic end of the pH gradient gel as you have seen in the figure. After applying an electric field, the proteins get separated depends upon its charge, either positive or negative charge. So based upon the pH of the surrounding medium, it will move and it will reaches and it gets separated at the specific point, that is called isoelectric point. Larger proteins will also move slowly through the gel, but that will take the sufficient time to catch up with the small proteins of equal charge. And now this is the second dimension, that the second dimension of this 2D electrophoresis is SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis. After the separation of this protein sample mixtures in the gradient gel, they get separated at the different points based on their isoelectric point. Then the samples are allowed to move in the gel, polyacrylamide gel. So before that, it will be treated with the STS in order to get the fractions, that is the polypeptide chains. So the second dimension is the separation performed by STS page, that is polyacrylamide gel electrophoresis, which is an electrophoretic method that is exclusively used for separating the polypeptides according to their molecular weight. This method often contains 4 steps, including the preparation of the gel, then the equilibrium of the immobilized pH gradient strips in SDS buffer, placing the equilibrated IPG strip on the SDS gel, and finally handling the electrophoresis. So this STS, the role of the STS is mainly exclusively for denaturing of the protein so that we can get polypeptides, and this will be helpful for the movement and of this polypeptide molecules. While applying this STS and the reducing agent, we can cleave the disulfide bond. So the disulfide bond which is present in between the protein, it will be acted upon by this SDS. So it gets unfolded into linear chain with a negative charge. So that is proportional to the polypeptide chain length. Polyacrylamide forms, the gel forms a mesh-like matrix into which that is appropriate for separating these proteins. So the proteins are separated by STS page. The smaller proteins migrate faster since they are having less resistance. And next step is the visualization of the proteins. There are various methods for visualization of the proteins, but the most commonly used thing is the staining with Kumasi blue as well as silver staining. So now we will see one by one. First one, silver staining. It is a sensitive and non-radioactive method. The principle is quite simple. The amino acid side chain can bind to the silver ions, primarily the sulfhydryl and carboxyl groups of these proteins, which is followed by reduction to free metallic silver. So as the result, the protein bands which are visualized as spots when the reduction occurs. Silver staining, it is suitable for low protein levels because of its sensitivity in very low nanogram range. The next one is the Kumasi blue staining method. It is relatively a simple method and also it is more quantitative than silver staining. It is suitable to detect protein bands containing about zero point 0.2 microgram or more proteins. The Kumasi blue, that is Kumasi dye, it binds to proteins to form a protein dye complex through van der Waals attraction. So there are two kinds of these dyes are used, that is R250 as well as G250. And now we will see the applications of this two-dimensional electrophoresis. It is extensively used for the quantification and analysis of proteins. So this method is exclusively used for protein source. It's a powerful technique for studying protein composition, modifications, as well as their interactions. It offers high resolution, reproducibility, as well as the ability to analyze complex protein extracts. Thank you.